Okay, as I told yesterday, today we will discuss the question based on respiration. Okay. Yesterday, of course, towards the end, almost 1 2 minutes before that, current was off. Okay, so internet connection was not there, so I had to stop it. So anyway, today we will do some question. Okay. Let us see that the first question related to biological in which dash is the most common. What is the options are here: respiratory substrate carbohydrate and respiratory substrate protein. Say this first word is for the first gap, and the second word is the second one. Okay, so you have to maintain the sequence also. In that way, you have to see. So the compound that are subject to biological oxidation is called what? Say respiratory substrate or respiratory substance. Here is a respiratory organic substance and respiratory substrate. Which one it will be? This is known as a respiratory substrate. Okay, in which there are respiratory substrate that we have learned. It is carbohydrate, then fat, protein, and organic acid. Is it organic acid? These are the respiratory, but the most common one is carbohydrate. So our this option that the respiratory substrate, the compound that are subject to biological oxidations are called respiratory substrate, in which carbohydrate is the most common one. Okay, so our answer is this one, the respiratory substrate and carbohydrate. Come to the next question. Site of glycolysis. This was asked in the Kerala 2011. This was asked. So here the site of glycolysis is, one is given cytoplasm, intermembrane space of mitochondria, mitochondrial matrix, and mitochondrial inner membrane. What is the site of glycolysis? Glycolysis takes place outside. It is outside mitochondria, is it? Outside mitochondria. So that outside mitochondria, it is the mitochondria are present in the cytoplasm. Okay, so here it is cytoplasm, but actual correct, a very correct word should be the cytosol. The site of glycolysis is cytosol. In the intermembrane space of mitochondria, that intermembrane space of mitochondria is the site where this proton, that is hydrogen ions, are accumulated in the mitochondrial matrix, is it? This is site for carbohydrate. Krebs cycle. And mitochondrial inner membrane, it is the site where that ETS, electron transport system, is located, and terminal oxidation takes place here. That electron carriers or electron chain, electron transport chains are located here. In the intermembrane space, this is location where this hydrogen ion are accumulated to develop the that proton pump. No, that proton gradient is developed. Proton gradient is developed in the intermembrane space, that is in between the two membrane, inner membrane and outer membrane. Okay, next question come. Respiration involves breaking of bond dash bonds of complex compound through oxidation within the cells, leading to release of considerable amount of energy. Say, this is the this one is that in respiration what happened? The respiration, the chemical energy that is released. All right, the chemical energy is released. How? The chemical energy are present within the bonds of the chemicals. Okay, that bond, and these our respiratory substrates are organic compounds. So in them there is the carbon molecules are there, and in between carbon molecules there are bond. So the bond between carbon bond, when this one break, that there is a release of energy. So this is respiration involves the breaking of CC bond. Okay, this CC bond, this bond breaks and release the energy. Not that CO or CH, mainly it is CC bond. These things you will get in your book, in the beginning, that introductory portion. Okay, there is a breakdown of the carbon bond. There's question 2, question number 3, that, and now come to question number 4. In both aerobic and anaerobic respiration, which of the following product is formed? Respiration is aerobic and anaerobic. The common product, the product out of options given here, and from this option which one is common for both type of respiration? See, lactic acid, option 1 is lactic acid. This lactic acid is formed in the lactic acid fermentation, and this is, that is anaerobic. This one is formed in lactic acid fermentation. Fermentation means it is anaerobic. Is it? The pyruvic acid is common because pyruvic acid is end product of glycolysis, and only after the formation of the pyruvic acid this it is decided whether it will go for the aerobic or for anaerobic. The glycolysis is common for both aerobic and anaerobic respiration. So end product of the glycolysis is common for aerobic and anaerobic respiration. So our answer is the pyruvic acid. This is the common product for aerobic. Citric acid is not formed. Other organic acid, those things, some organic acid will be mainly it is formed in the Krebs cycles, and other citric acid is the product in the Krebs cycle. Okay, and Krebs cycle is part of aerobic respiration. These are in the aerobic respiration only. Remember that in the aerobic respiration, in the Krebs cycle, the first compound that is produced, synthesized, this is a citric acid. And in the Krebs cycle, there are many organic acids are produced. Okay, so answer is the pyruvic acid. Next, question number 5. Fastest energy can be obtained from oxidation of which respiratory substrate? Very fast. It is the fastest. Say carbohydrate is the most easy thing to that is oxidized and digested. So it is the carbohydrate, fat, protein, organic acid. The primary thing is carbohydrate. If carbohydrate is not there, then only fat is used as a respiratory substrate. After that, protein and organic acid. So answer is carbohydrate. And that is why that during, if someone, okay, very weak person sometime get fainted, all right, then what do we give them? We give them glucose, is it? Or you can give them directly sugar, that our table sugar, which is sucrose, is it? And sucrose break down into glucose and fructose, all right, glucose and fructose, and they give instant energy because it is very fast. It is oxidized and give energy, glucose. So fastest is for carbohydrate, and if it is in the form of glucose, our energy drinks are usually glucose, though to glucon D, etc. is there. Is it? Next question. Question number 6. In order for glucose to be used as an energy source, when glucose is to be used an energy source, it is necessary that, what is the necessary condition? The necessary condition, glucose be formed from fructose? No. Glucose and fructose are isomers. First, glucose, usually what happen in the glycolysis, you have seen that first compound is the glucose. Then this glucose is converted into fructose. Is it? Glucose 6-phosphate convert into fructose. It is not that glucose is formed from fructose, not this one. Glucose be degraded to carbon dioxide. Glucose to be used as an energy source, this is also not necessary that it will. Here, if it has to be used for the energy source, this is two molecule of ATP be invested in the system. Do you remember that in case of glycolysis, in the process of glycolysis, what happened? That glucose is first converted into glucose 6 phosphate, that is ATP is consumed. Again, there is fructose. This one, glucose, it is converted into glucose 6 phosphate by the consumption of ATP. Again, there is another, this fructose 6 phosphate is converted into fructose diphosphate by the consumption of ATP molecule. So two molecule of ATP are consumed initially in the initial process of glycolysis. So the process of respiration start with the phosphorylation of the respiratory substrate. The glucose is phosphorylated. So two molecule of ATP are invested. It is first spent. Okay. So of two molecule of molecules of ATP be invested in the system first. First is you have to invest something for your earning. Whenever you are doing something, going to do something, you have to invest something. So energy is invested, ATP molecule. So our answer will be option number 3. Here is a question, a little bit different types. There is a passage with here is a passage with uh, dash space. Okay, so you have to fill this with the appropriate word from this option, right? The passage is, in glycolysis, glucose undergoes dash. This is A. Glucose undergoes what? To form two molecule of pyruvic acid. Okay. Glucose undergoes to form two molecule of pyruvic acid. In this glucose is derived from converted into C. You have to find out what is the C. And these two monosaccharides readily enter the glycolytic pathway. D is then the key product of glycolysis. D is the key product. Here in our option, what is there? Option number 1, this is A is oxidation, B galactose, C hexokinase, D oxygen. Number 2 option, reduction, sucrose, B sucrose, C glucokinase, D acetyl coenzyme A. In 3, partial oxidation, sucrose invertase pyruvic acid. And 4 is hydrolysis, maltose, gymase, pyruvate. So here the in glycolysis glucose undergoes what? Glucose is this is glucose is oxidized, is it? But this one is glycolysis. In glycolysis, the complete oxidation does not take place. Is it the complete oxidation? No, because complete oxidation takes place after glycolysis, then Krebs cycle, then only it is complete oxidation. So here it is that glucose undergoes partial oxidation, right? To form two molecule of pyruvic acid. In the glycolysis, it is glucose and it is two molecule of pyruvate or pyruvic acid. This is partial oxidation. In plant, this glucose is derived from what? Just I have told that glucose and fructose, it derived from sucrose, is it? Sucrose is a disaccharide, our table sugar, common sugar. Sucrose is a disaccharide and it is made of one molecule of glucose and one molecule of fructose. Okay, so this in plant glucose is derived from B should be sucrose, and these two monosaccharides by the enzyme C, the enzyme is invertase. Okay, this is enzyme which break down the disaccharides. This is invertase, and these two monosaccharides then enter the glycolytic pathway, glucose or fructose. D is the key product of glycolysis. The pyruvic acid is the key product of glycolysis. This is the end product. So answer is option 3. Next, phosphorylation of glucose during glycolysis is catalyzed by. Phosphorylation of glucose means here glucose is there and this one change into glucose 6 phosphate, glucose 6 phosphate, or you can say glucose monophosphate. Monophosphate 6 indicate the number of carbon, that is the phosphate group is attached to the 6th number of carbon. Glucose, fructose, they are 6 carbon compound. You have to remember this. And during this phosphorylation, there is consumption of ATP and production of ADP plus inorganic phosphate. But there is enzyme. This reaction is catalyzed by enzyme which is known as hexokinase. Kinase is the enzyme where there is a phosphorylation or involvement of ATP. That enzyme is usually called kinase. Okay. So it is hexokinase. Why? Because glucose is a hexose sugar. Right. So the name of the enzyme is hexokinase. So this phosphorylation process is carried out by, catalyzed by enzyme hexokinase. And here also metallic activator magnesium is also used as a catalytic activator. Okay. Next, the phosphorylation, next step also there is phosphorylation, glucose 6-phosphate convert into fructose 6-phosphate, 6-phosphate, then fructose 6-phosphate is changed into fructose 1,6 diphosphate. And this here also ATP is consumed, ADP is formed, and the enzyme here is this one also kinase enzyme, but we call this one as a phosphofructokinase because this kinase enzyme is changing the adding adding a phosphate group to the fructose. So it is known as not a hexokinase, it is specifically called phosphofructokinase. So these are kinase enzyme wherever there is phosphorylation is there. Next question. This is the number 7. This one is number 8. Now come to question number 9. Glycolysis occurs in the dash and produces dash which in the presence of oxygen enters the dash. So what will be the suitable word from this option? Cytosol, pyruvate, mitochondria. These are the words given, but their position has been, sequence has been changed in the different options. The glycolysis occur in the cytosol, is it? So our answer may be either 1 or 2, is it? So this mitochondria and chloroplast, number 3 and number 4 cannot be, because from the first dash, this one, it will be cytosol. And it produces what? It produces pyruvate. Here in the second option, it is written glucose. So no, in the glycolysis, start with glucose, end with pyruvate. So this option will be correct, that it forms the pyruvate, which in the presence of oxygen, if oxygen is there, that is aerobic respiration is there, then it will enter into the mitochondria where the Krebs cycle and terminal oxidation will take place. So our answer will be cytosol, pyruvate, and mitochondria. So option number one is our answer. Next question. Say this one is a little bit tricky. You have to remember the sequence of different intermediate product. You have to also remember the this abbreviated name of the intermediate compound. Can you tell what is that G6P indicate? Hello, anyone? We have G6P, PEP, PGAL, PGA. Can you tell this meaning? Anyone? Or whichever you can tell. Try.
Glucose 6 hydrogenase or glucose 6 phosphate hydrogenase.
Not hydrogenase. There is no hydrogen. No. This one is glucose 6 phosphate. Okay. Glucose 6 phosphate. Right. So this is glucose standing for G. G for glucose. 6 is 6. That is the number, which number of carbon to which this phosphate group is attached. So this is G. And what is PEP? PEP is PEP you have learned in case of photosynthesis also. Okay. Because see one thing that in the photosynthesis what happened, that glucose is synthesized, and in glycolysis glucose is broken down. So this process of the photosynthesis, no, in Calvin cycle, the chain by which the glucose fructose is produced, that is just a reversal of glycolysis. If the glycolysis go in the reverse direction, then it will be photosynthesis initial portion. That is the dark reaction, Calvin cycle. And of course, this PP we get in the photosynthesis in C4 cycle. That PP absorb carbon dioxide, is it? This is PP is phosphoenol pyruvic acid or phosphoenol pyruvate. Okay, PP, phosphoenol pyruvate or phosphoenol pyruvic acid. This is the last but one intermediate of the glycolysis. Phosphoenol pyruvate then change into pyruvic acid. This PGAL, what is PGAL?
Phosphoglyceraldehyde.
Yes, that is PGAL is 3 PGAL, that 3 phosphate group is on the third carbon compound. PGAL is phosphoglyceraldehyde. Glyceraldehyde. This is aldehyde. All right. And then PGA is produced in the photosynthesis also in Calvin cycle, first stable compound. And here in glycolysis also it is there. PGA is, okay, you tell who told about the PGAL. The similar thing.
Phosphoglyceric acid, sir.
Yes, yes, that's good. Phosphoglyceric acid. Phosphoglyceric acid. Okay. So, but now, see, this is what you have to know the abbreviated name also, or the alternative names you always because this question that they have not used the complete name of the compound. It is abbreviated, and if you don't know these abbreviated terms, then you cannot solve this question. If you know also, this is actually the sequence in the glycolysis. Okay, the correct sequence of the glycolysis. So you know the entire glycolysis, but if you don't know the abbreviated name, you cannot answer it. So what happened? Say it start with, say here, this glucose convert into glucose 6 phosphate, is it? Glucose 6 phosphate. This glucose 6 phosphate change into fructose 6 phosphate. Fructose 6 phosphate change into fructose diphosphate, and then this fructose diphosphate, say this fructose diphosphate, it will change into two molecule. This is the dihydroxyacetone phosphate, and another molecule is phosphoglyceraldehyde. Is it? These two are isomers. This PGL can change into dihydroacetone phosphate, and dihydroacetone phosphate can change into PGAL. This PGAL then change into PGA, phosphoglyceric acid. So in this chain, in that sequence, first in all, it is the first compound is glucose 6 phosphate. Second one will be PGAL, not PEP. PEP will be the last one here. And from PGAL, there is formation of PGA, and this PGA will form the PP, phosphoenol pyruvate, and this phosphoenol pyruvate, it will convert into pyruvate. Okay, so the last product. So this one you can see here. See this is the this one is the glycolysis. See here, this is glyceraldehyde 3 phosphate. Glyceraldehyde 3 phosphate is changed into phosphoglycerate or phosphoglyceric acid. Then there is formation of phosphoenol pyruvate, this one, and the end product is the pyruvate. So our sequence will be this one: glucose 6 phosphate, 3 PGAL to 3 PGA, 2 PP phosphoenol pyruvate, and end will be pyruvate. So question is asked in different way and you have to remember it. Next question is question number 11. The conversion of pyruvic acid to acetyl coenzyme A is a type of reaction called. What is the reaction called? You remember that after the pyruvic acid is formed, if oxygen is available, then it will enter, it will go to the Krebs cycle, is it? But in the Krebs cycle, the pyruvic acid is not directly enter into the Krebs cycle. But what happened? The pyruvic acid is changed into acetyl coenzyme A, is it? Pyruvic acid change into acetyl coenzyme A. So what is the reaction is called? This one. Who can answer? Anyone? Please try. Or see your notebook. Yesterday only it was taught. Which one will be the main? During the formation, here you see one this pyruvate. See in this question you can see this. This is Krebs cycle, all right, abbreviated one. In short, very simplified. This pyruvate change into acetyl coenzyme A, is it? And during this process, what happened? Carbon dioxide comes out, as well as hydrogen is removed. Remember, like this way, pyruvate, it changed to acetyl coenzyme A, and during this process, NAD involved, and with NADH2 is formed, and in this process also first molecule of carbon dioxide is released, and this reaction is catalyzed by pyruvate dehydrogenase complex. Enzyme is pyruvate dehydrogenase complex. So what is happening in this reaction, that carbon dioxide is released. So this one is known as decarboxylation. And this is dehydrogenation takes place. That a hydrogen is removed, and removal of hydrogen is called what? Oxidation. So this process, the process is known as oxidative decarboxylation. Because oxidation takes place and decarboxylation also takes place. So the answer is oxidative decarboxylation, option 3, not that oxidative dehydrogenation. Of course, it is dehydrogenation is taking place, but this reaction is known as oxidative decarboxylation. This oxidative at the same time dehydrogenation, it is the same thing because by dehydrogenation oxidation takes place. So here oxidative decarboxylation. Option number 3 in the question 11. Next one is, here is a question that we have to remember that different compounds, okay, that are formed in glycolysis, Krebs cycle, and those in the C3 cycle, C4 cycle in photosynthesis. What is the number of carbon atom in those compound? Okay, total number of carbon atom. So in this has been given the question number, here it is of course written question number 35. This is from the book from where the question has been taken. Otherwise you can mark it as the question number 12. Okay. This question, how many carbon atoms are there in S, T and U? This S has been uh, indicated, pyruvate has been indicated by S. Next one is here within the Krebs cycle. In the Krebs cycle, the compound oxaloacetic acid. This one has been indicated by U, and citrate, that is the citric acid. Citric acid. No, this Krebs cycle is also known as citric acid cycle. So this is citrate or citric acid. So this is T. So how many carbons in these compound? Options given like this: STU and 1, 2, 3, 4. First option, S is 3, T is 6, U 4. Option 2, 4, 6, 3. Option 3, 6, 3, 4. 4 is 6, 3, 3. What is the
364, sir.
364? S, T and U. S is 3. This is true. Okay. This is the end product of glycolysis. In the glycolysis, glucose and fructose, they are 6 carbon compound. And after that, whatever is the intermediate product, okay, all those are 3 carbon compound. And that is why from 6 carbon compound in the glycolysis, from 6 carbon, this break into 2 molecule of 3 carbon compound. This pyruvate is a 3 carbon compound. From here, see here, from this 3-carbon compound, 1 carbon is coming out. Then how many left? 2. So acetyl coenzyme A is a 2-carbon compound. Okay. The oxaloacetic acid, this one is a 4-carbon compound. So 4 plus 2. This there is a synthesis. This reaction, no, acetyl coenzyme A is accepted by oxaloacetic acid. This is a process of synthesis. This one combined together. So this is 4-carbon compound, and this one 4 plus 2 is 6-carbon compound. So here, so 4 or 3, it should start with 3. STU. See here, if you are confirmed, say if you know in this option, no, very easy option has been given. If you know only this one, that it is a 3-carbon compound, then you can answer it. Whether if you don't know this one or this one, but you can answer, because in this, for S, only this one, option 1 is having 3. Option 2, 3, 4, it is given 4, 6, 6. This is not true for pyruvic acid. So if you know, sometime this happen that the options are so easy that if you know one of them and you are confirmed, you are very confident, then you can answer it. So your answer is 3, 6, 4. This is 6-carbon compound, but after this alpha ketogluteric acid, this is a 5-carbon compound, because here during the formation of from citric acid to alpha ketogluteric acid, one molecule of carbon dioxide again come out. So this one is 5-carbon compound. I will show you. You can yes, you can see this Krebs cycle. Okay, and here you can see the cycle is clear. See here, pyruvate.
Yes, sir.
Can you see, no? The Krebs cycle, citric acid cycle. Say here, each
Yes, sir.
with each compound, they have written which one is how many carbon. 3-carbon compound. 1 carbon dioxide comes out, so 1 carbon is less. So acetyl coenzyme is 2-carbon compound. Oxaloacetic acid is a 4-carbon compound. And when this one oxaloacetic acid, acetyl coenzyme A combine together, then citric acid is formed, and citric acid is so 4 plus 2 is 6. After that, during the formation of alpha ketogluteric acid, 1 carbon dioxide is coming out. So it will be 6 minus 1. So this is alpha ketogluteric acid is a 5-carbon compound. Okay. After that, another carbon dioxide is coming out. Means one more carbon is less. So that's why it forms the, there is a formation of succinyl coenzyme A. Some all the intermediate compound has not been given here. See, this line is broken line, means this is the conversion of alpha ketogluteric acid to succinic acid is not a single step reaction. Okay, several step reactions are there. That's why broken line. Here there is no broken line. This is directly it is formed here. Okay, so there is a loss of one carbon atom here. So that's why this succinic acid is a 4 carbon compound. The succinic acid then change into, after that, the carbon dioxide is a release in these 2 3 steps. That's why after succinic acid, all are 4 carbon compound. There is fumaric acid is produced. From fumaric acid, malic acid to oxaloacetic acid. So these are 4 carbon compounds. So you have to remember all this, which one is, how many carbons are there. In glycolysis, there is 6 carbon compound and 3 carbon compound. Glucose, fructose, 6 carbon compound. After that, all those PGA, PGAL, phosphoenol pyruvic acid, all are 3 carbon compound. Okay. Next, come to the next question. In Krebs cycle, ATP the conversion of in Krebs cycle, ATP is formed directly during the conversion of what ATP is formed here. They have not this one. See, this GDP change into GTP. This GTP is converted into ATP. From the GTP, ATP is produced from ADP. All right. So it is directly, sometime it is written that in this place ATP is synthesized. So during the formation of what? ATP is directly formed from succinyl coenzyme A to succinic acid. Here, of course, this question is not it is not clear here in this cycle, because this is a simplified cycle. Some of the step has not been mentioned. Actually, here in this portion, there is a compound called succinyl coenzyme A. Okay, this is called succinyl coenzyme A, and this succinyl coenzyme A change into succinic acid, and when this happen, then ATP is produced directly. Succinyl coenzyme A to succinic acid. Here succinic acid to fumaric acid, during this step FADH2 is produced. Here also, this from the succinic acid there is fumaric acid. Fumaric acid to malic acid. From fumaric acid to malic acid, there is no formation of any NADH or FADH anything. Okay. So this is alpha ketogluteric acid to succinyl coenzyme A. No, this one, alpha ketogluteric acid to succinyl coenzyme A. There is a step here. Here is succinyl coenzyme A is right. During this, carbon dioxide and NADH2 is produced. So this question that in Krebs cycle it is directly formed from the succinyl coenzyme A to succinic acid. Wait, I will show you the detail. The yes, this one. This is the detail. This is the detail of the Krebs cycle. Okay, here say so many steps are there. No, citric acid to ceconitic acid. From the isocitric acid is converted into oxalosuccinic acid. Here NADH2 is produced. Oxalo Oxalosuccinic acid is converted into alpha ketogluteric acid. Here the carbon dioxide is produced. You can see here carbon dioxide. This carbon dioxide you can see here. It is produced oxalosuccinic acid to alpha ketogluteric. Alpha ketogluteric acid is converted into succinyl coenzyme A. And during this process, carbon dioxide and NADH2 is produced. Succinyl coenzyme A is changed into succinic acid. During that period, a GTP is produced. So succinyl coenzyme A to succinic acid. And when succinic acid is changed into fumaric acid, there is formation of FADH2. This photograph is not very clear. Okay, this is FADH2 from FAD. And then after that, fumaric acid to malic acid, there is no formation of any reduced coenzyme. After that, this one is malic acid. Malic acid is converted into oxaloacetic acid. Here NADH2 is produced. Okay, next question 12. This one is question number 13. Next question, it is question number 14. Pyruvate dehydrogenase is used to convert pyruvate dehydrogenase is pyruvate is changed into acid. Enzyme is pyruvate dehydrogenase. Okay.